So we're going to start by talking very briefly about your sensory receptors, and then we'll do the beginning of photoreception, but it'll take us a little while to talk about all of the details that go along with your eyes. So we finished chapter 11 with lesson 5, which was the structures of your central nervous system. So this is lesson 6. And we're going to start just with a little bit of brief description about some words that you will see when we're talking about sensory receptors and sensation and perception. The first important thing is, what is a sensory receptor? So a sensory receptor is some kind of specialized cell. And there's different kinds, and I'll give you some of the categories that exist today. But it's a specialized cell that detects a stimulus in the environment. And like we already saw in our last set of lessons, the sensory receptor would be attached to a sensory neuron. Your sensory neuron usually is not all by itself. So we know from our lessons on organization of your nervous system that a whole bunch of neurons are probably going to go together to make a nerve. And this nerve is going to go somewhere in your central nervous system. Now, the pathway would continue if it was a reflex arc, but we're going to talk about perception, not just automatic reflexes. So now, where we go in the central nervous system will be a little bit different, and it'll change depending on the sense that we're talking about. So I'm just going to end the pathway there for now, and when we talk about each of the senses, I'll tell you where it's going. So there are two words that often get used together, sensation and perception. Sensation is the detection of a stimulus, which your body does sometimes without you being aware that it is happening. And that is because perception is the part where you become aware of the sensation. So perception is the interpretation of a stimulus. Sensation is accomplished by the receptor and the nerves. Interpretation or perception, that's what your central nervous system does. So there are plenty of times where your nervous system senses something, but you don't perceive it. And that is because sensory adaptation can happen. In our class last week, where I made you look at a bunch of different things randomly, we did a bunch of sensory adaptation, where we tricked your brain into thinking that either something was there that wasn't, or something wasn't there that actually was. In sensory adaptation, essentially, unimportant information is filtered or ignored. So for example, I can hear, if I pay attention to it, the projector for my smartboard kind of making a whirring sound. It's kind of buzzing. But it's not really important, so most of the time, I'm not even aware that it's happening. Even though my ears are totally detecting that stimulus, my central nervous system is saying, yep, we hear it, but that's not really important for right now, so we're just going to ignore it. I showed you guys all how you didn't hear the clicking of the clock until we stopped, focused on it, and listened, because whether or not the second hand is clicking wasn't super important to your survival at that point in time. So the key point here is that it's unimportant, and what is unimportant can change. So at some points, it might be incredibly important for you to know exactly how many seconds have gone by, but at other points in time, it probably isn't. I always like to remind people, and I would have mentioned this last week, the feeling that your clothes cause against your skin. I assume that you are wearing clothes most of the time, but you are not acutely aware of how your clothes feel touching your skin, because your brain is like, yep, okay, clothes are there, feel them, but don't really care, because it's not really changing anything for what I'm doing at this point in time. Now, does anyone want to ask any questions about any of these basic words? Oh, it must have been funny because the people were laughing back there. Let's go, share it up. No, he doesn't want to now. I've embarrassed him. I activated his sympathetic nervous system just there. So you have a whole bunch of types of sensory receptors, and we're going to group them into categories. One category of receptors that you have are called chemoreceptors. So that implies that there is some chemical molecule that is being detected. Now, 3 examples of places where you have chemoreceptors would be for taste, for smell, and for internal conditions. So that you know, the details of taste and smell and internal condition receptors are not something that is tested. You don't have to know the structures and all of the parts. I just want to show them to you as examples because I think it's important for you to know that they exist. So on the left here, we have some information about taste. No one will ask you this on a test, but on your tongue, different areas of your tongue are sensitive to different tastes. You guys can shut the drawer if you want, if that's bugging you. Are we okay? There's different tastes, so that implies that probably you have different thresholds. Because different parts of your tongue respond better or worse to different tastes. I think everyone's probably clear on what sweet and salty and sour are like. Bitter is like what soap would taste like in your mouth. And umami is it's called savory. Meat is the way I would describe that, the way meat tastes, good and savory, or maybe gross if you don't like the taste of it. That's the umami one. You won't get quizzed, like no diploma question is going to ask you to have memorized where different tastes are on your tongue, but I want you to recognize that you taste different things because different neurons are activated, and if different neurons are activated, your brain gets a different signal. So you have different taste thresholds, and you have different neurons activated. When we're talking about taste, remember all sensation starts with a sensory receptor that goes to a sensory neuron. In this case, it is the parietal lobe of your brain that is going to help interpret taste. Now, some people would argue that taste and smell kind of go together, and don't forget your brain is not these individual packets that each do their own thing, they work together. So very often, what you taste and smell sort of enhance each other. I know my husband will regularly say if he's got a cold and he's stuffy in his nose, he doesn't feel like he can taste the food that he's eating because those parts of your brain kind of work together and they take the stimulus from both of them to make this enhanced perception of what's happening. This is what it looks like on your tongue. You have a pore. Inside the pore are specialized cells. Those are your taste receptors. And then there is neurons that go together to make a nerve. If we're talking about smell, at least one group had the olfactory bulb in here. Who had an olfactory bulb on their brain? Someone did. And I was like, oh, I'm so excited for you. You have the olfactory bulb still attached. It was this structure on the dissection of the brain where I said, if you can see this, cool, but a lot of the times it's not attached anymore. So if we're looking at where this is, there's the frontal lobe, and right underneath it is where you should see the olfactory bulb. So it's this little structure here. It's attached to this whole other part of your brain that has to do with how you feel, whether you're happy or scared or sad, called your limbic system, that we don't even enter into discussions about. So if we're talking about smell, still chemoreceptors, right? So it's still molecules. But this time, the signals are going to your frontal lobe instead of to your parietal lobe. So just like with taste receptors, there are receptors on the olfactory bulb that are attached to neurons, that are attached to nerves, that go to your frontal lobe, and that also go to this place called the limbic system. So you will not have to, on a test, label structures of your nose or label taste buds on your tongue. You will have to understand that both of them are chemoreceptors, so they detect a molecule which stimulates a receptor, which is a cell attached to a neuron that sends sensory neurons to the central nervous system. There are other types of receptors that are harder to draw pictures of sometimes that are for internal conditions. And so I'm just going to give you an example here of one that you have probably heard of. If you took Bio20, you should have talked about these or what they do. Osmoreceptors. Does anybody remember what they are? So you need to maintain water balance in your body. And so you have receptors in your nervous system as well as inside of your blood vessels and a bunch of other places that will help to detect how much water there is. If there is a lot of stimulation of the osmoreceptors, that tells your body, oh, I need to reabsorb water or I need to let water out. So osmoreceptors are for water balance. There are other receptors. You have chemoreceptors in the medulla of your brain. They help manage CO2 levels. So these chemoreceptors detect, really what they're detecting is higher levels of hydrogen ion concentration, which is what happens when CO2 dissolves in your blood. And that will help adjust your respiration rate so that you can have less CO2 and more oxygen. Does anyone want to ask anything about any types of chemoreceptors? Yes.
So your internal conditions just basically like regulate stuff in your body, so like if it's trying to balance water, like it would be like thirsty or something.
Exactly. So a part of the osmoreceptors pathway would be to do automatic things like tell your kidneys to reabsorb water, but it would also stimulate you consciously to say, hey, you know what? I am thirsty. I should go drink something. So that would be the conscious part of your central nervous system that's activated. You also have what are called mechanoreceptors. So in the mechano part of the word, it means that something is physically moving or being pushed on, but usually moving. Touch, pressure, and pain are all felt in your skin. There's a whole bunch of different types of cells in your skin whose names you do not have to memorize. But I put a little picture there to illustrate all of the stuff that's going on under your skin. The top of the picture is your skin, and you can see little hairs coming out. And as you go down from the surface of your skin to the more internal layers, you see a whole bunch of different types of receptors. So there's free nerve endings. Those ones are the ones that are sensitive to hot, cold, pain. So when I asked people to put their hands in hot or cold water, I was activating those. And because they respond to both hot and cold, your brain was confused about which one they were actually detecting. There's a bunch of different ones that are for touch, like a Merkel disc or a Krauss end bulb or a root hair plexus. You don't have to know those names, but there are a bunch of specialized cells that are for touch. Over here, we have a couple that are for pressure. And so you have all of these different types of cells in your skin, each of which can detect different levels or varying degrees of touch or pressure or hot or cold. Okay? So you have your skin receptors. If I was to ask which part of the brain is this related to, we would say mostly your parietal lobe, because parietal lobe is for sensory information. So all of that touch and pressure stuff would go to the parietal lobe. If we consider hearing, most people say hearing, how on earth is that mechanical? But in hearing, sound waves cause vibrations. So when sound enters your ears and is detected by the receptors there, it causes things to physically move. So hearing has to do with your ear, and then it would go on to your temporal lobe, right? So that's where auditory information is analyzed. Balance is similar to this in the sense that it is also your ear. So your ear does two things. Your ear helps you hear, but your ear also helps you balance. When you move, any sort of movement shifts ear, fluid, and structures. So when you are, say, at an amusement park riding one of those crazy rides, which I don't do, so I can't tell you what it feels like because I've done it a few times, I'm never doing it again. What's happening is the fluid inside of your ear and little tiny things that almost look like rocks slosh around, and that tells your nervous system, hey, this person is moving. Now, because this is a little more than just sensation, it won't just be your parietal or temporal lobe. So I'm not going to put one specific brain lobe here because it's kind of hard to map them just to one. The last one is your body position. So we're talking about muscles or ligaments here. Muscles or ligaments have little receptors in them that basically indicate if they're stretched or not. And so you can detect, even if you can't see, so even if I close my eyes, but I put my arms out like this, I know that my arms are out here at my sides, up in the air, and not down in front of me because I have receptors telling my nervous system, this is where your body parts are. It is called proprioception. It's kind of a long, fancy word. It means body position. Now you also have things that are specifically thermoreceptors. So this is for hot, cold, warm, or anything in between. These are found in your skin along with all the touch, pressure, and pain receptors. And then lastly, you have photoreceptors. Photo means light. So these are light receptors. So these are in your eye. And the occipital lobe would be the first place that that kind of information is analyzed. So there's a bunch of kinds of receptors. You should have a good idea of like what kind of things would fall into each category. And if I told you which part of the brain is attached, which part of the brain should that information go to? That's the basics. For these three specific things, hearing, balance, and photoreception, we'll do a more detailed account of what's going on. So for example, for touch, pressure, pain, you don't have to know the names of those receptors or any details other than they're mechanoreceptors in your skin that send information to the parietal lobe. If we're talking about hearing or balance or photoreceptors, we will do the next 3 days of lessons just about those things, and you will have to know specific parts. So I'm going to start talking about your eye from the outside in. Okay? If we look at the outside of your eye, I'm going to point out in what, in my opinion, the least important structures are first. For example, I have never seen a diploma question that asks you what your eyelid is or about your eyelash, even though they are super important for protecting your eye because they don't play a humongous role in how vision actually happens other than they either open or close your eye. I don't see them get asked about. So they're there, they protect your eye, but I doubt that they'll be the subject of a question. Another structure that, although is important, isn't one that's listed that you specifically have to know about, which means If they were going to ask you something about the conjunctiva, they would have to tell you what it is and give you some context. The conjunctiva is near the bottom of your eye. It's really important in terms of tear formation and pressure in your eye. But those three structures at the front of your eye, I wouldn't expect you to have to memorize the function for. The other thing is over here, you have some muscles attached to your eye that helps you move your eye like up and down and back and forth so you can look at different things. So while those structures are there, and they are important, and they are often shown in pictures, I wouldn't expect what's the function of the rectus muscle to be a diploma question. Now, if we consider the outside of your eye first, and I'll try to color code this as best I can, we're going to start by talking about the sclera. So the sclera is this line that's pointing to the yellowy beige colored outer layer of the eyeball. When we dissect an eyeball, it looks pretty white. Like, to me, it doesn't really look that yellow. It looks pretty white, maybe a little bit on the off-white side. But the sclera is there, and its job is to support and to protect your eye. Now, if you follow that layer, it goes around most of your eye. So it starts right here, and then it goes all the way around your eye, except for that one spot in the back. So most of your eye is surrounded by this layer, the sclera. But as you move towards the front of your eye, this layer becomes something else. If I continue right here, that's your cornea. Your cornea, while it can help protect your eye, that's not its main job. The function of your cornea is to direct light into your eye. And if your cornea is misshapen, it can lead to vision problems. So I had LASIK done, and my cornea is what they changed. Essentially, in the procedure I had, they lifted my cornea up, used lasers to reshape the inside, and then flapped it back down. Took about 8 minutes, and then I could see. It was terrifying / fantastic. I didn't want to do it. Even till like the minute I was there, I was like, these are my eyes. What if they screw up? I'm going to be blind. But then after it was done, I was like, wow, why did I wait so long to do this? This is amazing. But your cornea is really important, not just for protecting your eye, but for helping you see clearly. If any of you have taken physics and talked about refraction of light and how light bends as it meets different mediums, your cornea helps with that. It helps direct light into your eye. So these are the structures that are on the outside. The sclera, which goes kind of all the way around, and then at the front, the cornea. If we move to the next layer, so if we're going from the outside in, the next layer, I could start by talking about this dark blue layer, which is the choroid. It goes in basically the same place the sclera goes, but it's one layer closer to the inside. It is dark on this picture, and that's how it is in real life. It's so dark that it looks black, actually, when you look at it. So the choroid, I'll just make a note here. It's so dark that it looks black. It absorbs light. One of the things that's important is that there's not random light rays reflecting all over the inside of your eye. So it's important that you have this dark thing to collect all of the excess light so that only your photoreceptors are actually detecting light. But this is also where the blood supply is. So if we were to ask you anything about what blood does, blood transports things. That's how your eye is getting oxygen. If something were to be damaged in the choroid, then it would inhibit blood supply to the cells of your eye, and so they wouldn't be able to get glucose, oxygen, or get rid of waste products. Okay. If we follow the choroid around, kind of like we did with the sclera, and we come towards the front of the eye, we don't call it the choroid anymore. There's a little layer It's this layer that's kind of a little bump here and it has some dots in it. And there's two of them, one on the top and one on the bottom, but only the one on the top is labeled. That's the ciliary body. And you'll notice that the ciliary body is attached to what looks like red fuzz. It's trying to mimic the look of a ligament. Ligaments are the things that help with your muscles and other places as well, so not just in your eye. But the ciliary body is attached to some ligaments. And so typically we would talk about those two structures sort of together. Now the thing that's in between them is the lens of your eye. And one of the things that we'll see is that for you to see things that are close to you, that are far from you, that are at a medium distance, your lens needs to move and change. And so your ciliary body and your suspensory ligaments, those ones help to adjust the shape of your lens so that you can see things more clearly. So I will just now in the next lesson where we talk about specific ways that things happen, I'll give you a bit more detail about this, but the word is accommodation. One of the things that gets tougher as you get older is moving your eyes from close to far, and it sometimes takes time for your eyes to adjust, or the ligaments or the ciliary body have weakened and it's hard for your eyes to even adjust at all. So if you have heard of reading glasses and that some people need them as they get older, this could be the reason, is that you cannot change the shape of your lens as effectively. Now, kind of coming from that same section are the next couple of pieces. This piece that sticks out here on the top and on the bottom is your iris. That's the color. So whatever color your eyes are, it is because the iris of your eyes. But that's not all it does. It's not just there to be pretty. The iris is the color, and it also adjusts your pupil. So when we did the reflex lab and you went in the dark room and then flashed a light in your eyes and your pupil changed size, it was because the iris actually changed size, adjusting the size of your pupil. Okay? And then the pupil is the opening here for light. So your iris and your pupil work together to adjust how much light is coming into your eye. So on the outside, sclera and cornea. Middle layer, choroid, which kind of flows into ciliary body and ligaments, as well as your pupil and your iris. You'll notice that there are some chambers, so we'll just highlight them. Right here, the aqueous chamber, and over here, the vitreous humor. Those two structures kind of have a similar function. So this one and this one. The fluid that's in here does things like maintain shape and pressure. If there's too much of the fluid, it can put too much pressure on the eye and it can cause pain. And this fluid is thicker than water. So when we do the eye dissection, you'll see it's more like jellyish than it is like water, but it's still a fluid. And because it's a fluid, things can get dissolved in it. And so sometimes people will have conditions where there's something wrong with the contents of these fluids in their eye. Now, as we move from the outside to the inside, we've been talking about there's an opening for light, there's things that change in response to light, there's things that direct light into your eye. And they're all trying to get light to directly contact the lens, which is this big round structure here. The lens's job, and I'll put the lens's job down here, is to refract light. The material that the lens is made out of has a refraction index that should, if your lens is the correct shape, direct light exactly onto a particular spot of your retina. So we'll see if my art skills are good here. If you look from the middle of the lens and you go exactly perpendicular to that, that is where light should be refracted. That is the goal of your lens. Now, some people have a misshapen lens. Some people have an eye that's the wrong shape for the lens that they have, and it might make them see things incorrectly. But ideally, when light comes and gets directed into your eye by your cornea, your iris adjusts the size of your pupil so that light can enter and it hits your lens, it should be refracted directly onto that back part of your eye. So your lens's job is to refract light, and its job is to refract light onto the retina. So the retina is this lighter colored inner layer here. And so typically, because questions are not usually printed in color, you won't see colors, you'll just see 3 layers of tissue. The outer layer is the sclera, the middle layer is the choroid, and the inner layer is your retina. Your retina contains photoreceptors. So this is where the specialized cells that actually detect light and are attached to neurons are found. Where light is supposed to be directed is called the fovea, and the fovea is just a part of the retina, so it's one specific section of the retina. The fovea has, or it should have, the highest concentration of photoreceptors. So if I were to map out your retina, most of the photoreceptors will be in this middle part where the fovea is. Now, you'll notice, and I specifically did this, I made my line pause and stop at the blind spot. The blind spot is an area where there are no photoreceptors. And it's because the optic nerve, which is the nerve that is going to go to your brain from your eye, has to leave somewhere. And it has to leave your eye. So there needs to be a space for all of those neurons that are coming from all of the photoreceptors to meet up and all leave the eye at the same place. And we tested your blind spot. It was the one where you had to look at the cross with one eye and eventually the little dot disappeared. That was us showing you where your blind spot is.